Awesome. So welcome everyone. I know folks are still trickling in, but I think it's time for us to get started. Before I get started, is everyone able to see my presentation and also hear me okay?
Yes.
Awesome. Thank you. So hi everyone. Welcome to our second Benchling Bootcamp session. This is in our series on the notebook application, and today we're going to be diving into some advanced features. So to start us off with an agenda, we're going to welcome everyone who's here on this webinar, as well as the folks on the Benchling team that are here to support you today. We'll then dive right into an infrastructure and overview of the Benchling platform. We'll then spend some time giving a refresher of some of the basics that we covered in our last Benchling Bootcamp session. We'll then be spending the majority of our time within the Benchling platform, going through some advanced notebook exercises. And then to wrap us up, we're going to connect everyone with recent product updates and show you where you can access those. We'll also be having a survey at the end of this training session, so do watch out for that. We do, of course, appreciate your feedback on this bootcamp, and we'll also connect you all with next steps. So here, these are the folks from Benchling that are on our call today. My name is Alex Chow, and I'm a customer success specialist with Benchling. We also have Chin, Stefan, and Meredith, as well as Freeman. So these are all folks from the customer experience team here at Benchling. So I'm going to be going through today's exercises and walking through the webinar, but these folks will be supporting your questions during this webinar. So we're all here to support, and we're excited to meet all of you. So just to set expectations in terms of today's webinar, we are of course using Zoom, and we will be leveraging some of the key features that this platform offers. In particular, if you do want to ask any questions during today's webinar, please take advantage of the Q&A button shown by this Q&A icon at the bottom of your Zoom screen. This is the primary way that you're going to be interfacing with us during the session. So as you ask any questions, excuse me, one of the other Benchling panelists will answer your questions promptly. Importantly, we're deciding to streamline our communications with attendees, and for that reason, we ask everyone to use the Q&A, and we've disabled the chat feature for today's webinar. Also to call out, we are going to be utilizing Zoom polls, similarly to how we used them for our last session, so do watch out for those. Those will appear as a pop-up window on your screen, and I will also verbally go through them as well. And these are just a way to re-engage with the audience and gauge everyone's understanding. So do watch out for those as well. Wonderful. So we're going to dive right into the Benchling platform. Again, when we talk about Benchling, Benchling is based off of these 7 applications. And again, our first Benchling Bootcamp series is based around the notebook application. And I'm sure, as you'll remember from our Benchling Notebook basic session, we discussed how the notebook is really a central source of truth within Benchling. It is highly integrated with the other applications on our platform. And importantly, it's a place for you to utilize notebook entries as both observational records and also as experimental protocols. In addition to these applications, our data model captures information or allows you to capture structured information about biological entities that you may be working in the lab, such as DNA sequences and protein sequences or amino acid sequences. And importantly, we discussed the main component of the data model that lives within the notebook, and this is notebook entries. So we'll now go through a refresher on some of the concepts that we covered in the Benchling Notebook Basics Bootcamp that happened a couple of weeks ago. So again, to give everyone a reminder of project folder structure, within the Benchling Notebook, we do have a hierarchical organization of the data that lives within it. So to start from the bottom, the data that populates the notebook application is called a notebook entry. And again, notebook entries are essentially dynamic text editing files in which you're able to plan your experiments, execute research, and also record your observations, and also attach external data files. So this is the piece of data that lives within the notebook. Moving up from entries, we then have folders. And folders are simply a way for you to Add additional organization to the notebook. Importantly, they do not set permissions. Again, they're just a way to organize the data within the notebook. The highest level of organization within the notebook is called projects. So a project is a way for you to set permissions for the data within the notebook. So importantly, any notebook entries that live within a project will gain their permissions from that project. So there are multiple ways that groups that use Benchling can organize their projects. Some groups may organize them by research group or target, or also by an individual working for that organization. And again, as a best practice, we generally recommend minimizing the number of nested folders. As I'll be demoing today, we have a powerful global search application within Benchling that gives you a lot of precision to be able to find your notebook entries as well as any of the other data that you're working with on Benchling. So we really see searching as the primary way that folks are able to find their data as opposed to a complicated folder structure. So now, as a quick refresher of some of the navigation that we covered in our basic session, to start us off, we have the Benchling toolbar or the left side navigation bar. And this is the bar in blue that's always going to be located on the left side of the Benchling screen. And in particular, this navigation bar is populated with a few icons in white that correspond to different functions and applications on Benchling. And we went over in particular the home application, the projects application, which is where the notebook lives, as well as the global search and global create. And importantly, we also touched on this personal avatar, which is located at the bottom of the navigation bar. So it's always going to be in the bottom left of the screen. And when you click on your personal avatar, which will have either your personal initials or a photo if you choose to use one on Benchling, this will pop up the user avatar and feature settings. And from here, you're going to be able to access your personal profile settings. If, for example, you need to change your email or the email that's associated with your Benchling account, you can do so. And you'll also be able to access a number of other Benchling settings. And I'll be showing, for example, how we're able to access notebook template settings from this same avatar menu. We also went over how we can access project settings from this gear icon located next to the name of the project that you're working with. And we also discussed how notebook entries have specific icons that allow you to toggle on different views. For example, this gear icon, which will always be in the upper right of your notebook entry, will allow you to toggle on several different features, including the dynamic table of contents, as well as timestamps. Wonderful. So that's our content in terms of the slides. I'll now transition to the Benchling platform. Wonderful. So we're now looking at the Benchling platform. I'm here on this dedicated training tenant that we've spun up for our Benchling Bootcamp exercises. So to get us started, I'm going to navigate to a notebook entry. And as I'm sure everyone remembers from our basic session, in order to come to an entry, we're first going to navigate to our project. So of course, the projects application is marked by this briefcase icon, again, in that left side navigation bar. So I'll click on projects. And as we can see here, I've already opened up this bacterial toxin detection, but just as a quick review, I can follow the breadcrumbs navigation to go back to see all of the projects that I have access to by clicking here where it says projects. And from here, I can see a view of the different projects that I'm able to access. So again, I'll click on this bacterial toxin detection project. And I'm now able to see any of the data, including notebook entries that lives within this project. So for today's example, I'm going to dive back into that same notebook entry that we used for our basic session. And I'll just close out of the left side navigation bar to give myself a little bit more space. And I can do so by hovering over the right facing caret and then bringing the cursor down to this X. So I now have a larger workspace to view my notebook entry. So as a quick refresher from our exercises during the basic session, we were focused on a particular research area, which was infectious disease. And in particular, we were focused in on research focusing on anthrax. So for our particular experimental design, we were using this model where we inoculated a animal model, in this case mice, with the bacteria that causes anthrax. We then collected a variety of tissue samples from these animals, and we then performed an indirect ELISA on these samples. In particular, our target for this ELISA was looking for the concentration of the anthrax toxin within these samples, because for us, this is a readout of the severity of disease in this particular model. So again, we'd like to hone in on a particular scientific use case to make this as engaging as possible, but again, want to emphasize that the exercises that we're going to be going through today, similar to last time, certainly apply broadly, as we understand our customers come from a diverse array of different life sciences research applications. So just as a review of some of the activities that we performed with this entry, we showed how we have built out these different steps for us to both put down observational notes as well as to look at the different steps that we have in our protocol. So now to fast forward to the end of this entry, we finished off our entry by attaching the raw Excel file that we got from the plate reader once we had finished our ELISA. So I'll review that workflow again. So when I want to insert a data file, I can simply click to where I'd like to insert that file. I'll then come to the Insert button, which is located in this formatting toolbar. And from the resulting dropdown, I'll come down to Attachment, shown by this paperclip icon. And here I get this resulting window where I'm able to upload a file from my device. So in this case, I will drag and drop my data. Here I have my ELISA raw data on my personal device. So I'll just drag and drop this over to this Insert Attachment window. And I've now uploaded my ELISA data. So I can preview this file by double-clicking on this attachment. And this will open a new sub-tab for this particular attachment. And I'm able to scroll through this. And as you can see, this Excel file captures some metadata from this particular plate reader, as well as a table with the raw values from my ELISA. So now to jump into our exercises, again, we showed how we're able to attach our raw data that resulted from our experiments. But let's say, for example, there's something interesting about this data. Maybe there was something wrong with this ELISA. Maybe potentially there were some edge effects on the edges of our plate, and we'd like a colleague to take a look at this. So we understand that there are many use cases for sending data back and forth between members of your organization. So that brings me to my first poll question. So to start us off with our first poll, our question is, how often would you send files via email or other to your colleagues? And the options are very often, often, rarely, or never. So do take some time to fill out this poll.
And Alex, after the poll is done, do you mind showing how to attach that raw Excel one more time? There's a question from the audience to.
Yep, absolutely. Thanks for calling that out. Wonderful. So it looks like most folks have voted. So I'm going to end this poll and I will share the results. So it looks like many folks, in fact, 46% of you said you send files via email often. And many of you also said you send this very often. So given this, we understand that there are multiple pain points with sending data via email. So hopefully show how we're able to utilize a feature on Benchling called the desktop sync application to mitigate some of these pain points. So again, to walk through how we're able to upload that raw data to start off this exercise, I'll start off by deleting this existing attachment. So I'll just click on this attachment and click delete. And from here, I'll show everyone again how I'm able to insert the file. So I'll again go back to that insert button in the formatting toolbar and click on attachment. And then from here, I'm just going to drag and drop from my desktop. And you, of course, can also click on upload a file and navigate to that file within your device. So again, I'm just going to come to my desktop, and drag this raw data and drop it right on top of where it says upload a file. And Benchling has now uploaded my ELIZA raw data into this notebook entry. So to bring us back to this main question, which is on sending data back and forth, we understand that there are a number of different use cases. Additionally, we understand that when you're sending data back and forth via email, there's an issue of source of truth for your data. Essentially, folks are editing a piece of data and there are multiple, or if folks are editing data that's being sent over email on their personal device, we understand that this can create a high number of versions of your data. I certainly know I experienced this in my time as a researcher. We, for example, had spreadsheets where we captured information on different animals that we were using in the laboratory. Multiple folks would make edits to this Excel spreadsheet and then not share these edits, or we would end up having different versions of the Excel sheet depending on who was editing this. So essentially there wasn't a central source of truth for our spreadsheet, and this caused miscommunication as well as other issues with maintaining this data. So to mitigate this, we offer the desktop sync application. So again, we have this raw data that's located within our notebook entry, and I can edit this data such that it will always be updated and it'll maintain its version within the notebook entry. So to do so, I'm going to click on this floppy disk icon at the attachment, and you'll see that I get this pop-out that says checkout file. So I'll click. And what this does is this triggers the desktop sync application. So importantly, I already have the desktop sync application downloaded, but if you haven't downloaded this already, we will provide a link or you will be prompted to download this from the download page. Importantly, that will be specific to the Benchling tenant that your company uses. So I've now checked out this file. What this does is it locks this external data file for editing. So essentially, if you'll remember from our session last time, we are able to add multiple co-authors to a single notebook entry. And as you can imagine, if we have folks editing the attachments simultaneously, we can end up with that same issue of different versions and a lack of source of truth for our external files. So for this reason, we allow you to check out a file which locks out any of the other authors from performing edits. So now that I've checked out my file, I will click Edit. And I'll allow Benchling to open Benchling Sync. And what this has now done is it's opened my ELIZA raw data in the native application on my device, which in this case is Microsoft Excel. So from here, I'm able to see my raw data. And let's say, for example, I'd like to create a visualization of some of my data. So I will highlight this, and you'll forgive me, I'm much more trained in Benchling than I am with Microsoft Office, but let's say we would like to insert a bar graph. Wonderful. So we've now created a bar graph of our ELIZA data. So I'm now going to save my edited file attachment within the native application, and I'll close out of this. And we can see that Benchling is syncing up my external file with the attachment that lives within Benchling. Importantly, I do want to call out that you saw that I closed out of my Excel window. We do recommend this as a best practice because once we've checked the file back in, the edits that you perform in the native application will no longer be synced up with the attachment in the notebook entry. So again, as a best practice, once you've finished any of the edits you need to make, save the file within your native application and then simply close out of it. So now that I performed the edits that I need to make, I'll now check this file back in. This means that the file will now be editable by any of the other co-authors on this entry. So to do so, I'll click where it says checked out. And we can see that Benchling confirms that the file was just synced. And I will now check in. Wonderful. So we've now checked the file out and back in. The external file is now available for edit by any of the co-authors. And I do want to show that this attachment now has this bar graph added to it. So I'll double click on this file attachment. And you can see here within this preview sub tab that I'm able to see the bar graph that we added to this particular ELIZA data. Wonderful. So we showed a few features of the desktop sync application with regards to adding a bar graph to Excel data. We'll dive now into how we're able to add excerpts from image-based file attachments, such as PowerPoints, as well as PDFs. So let's say, for example, in addition to performing these ELISAs for the anthrax toxin within animal samples, let's say as a part of our experimental design, we're also performing genotyping on these animals as we hypothesize that the genotypes of these animals may confer resistance to the anthrax illness. So let's say we'd like to attach our genotyping results, which in this case may take the form of gel images. So I'll again insert a new file by coming to the Insert button in the formatting toolbar and clicking on Attachment. And I'll again drag and drop the file from my computer. In this case, I'll use this PowerPoint file. So again, I'll just drag and drop this to where it says Upload a file. Wonderful. So I've now attached my PowerPoint. Importantly, we may want to add excerpts. So we are able to add excerpts for this attachment because we have this leaf icon next to the attachment. Importantly, excerpts are supported for, again, some of those image-based file attachments such as PowerPoints. So I'll click on the insert excerpts icon shown by this leaf. I'll then be given the option to select the slides of my PowerPoint as excerpts. I also can select these directly, although this does take a minute to load as PowerPoints are somewhat large files. So let's say, for example, I know that I'd like to show the contents of slide 2. I can type in the page numbers. And it looks like this is taking a minute to load, so I'll just repeat this action. Again, I'll click on the Insert Excerpts. There we go. So I'm able to either click on the pages that I'd like inserted. So let's say, for example, I know that this is the gel image that I'd like to insert. And then I can click on Insert. And what this has done is this has created an excerpt of a particular slide in my PowerPoint such that it shows up as an image within my notebook entry. So we understand that, for example, you may have PowerPoints or other image-based data files that are very long. For example, I was performing genotyping on many, many animals. I had many different experimental conditions, and I had 50 different gel images that I'd like to attach. But let's say in particular, I'd like to focus on just one of those images. You're therefore able to leverage excerpts as a way to have a preview of just one particular slide or image from that file attachment. So now that I've added the external data, I've created a visualization of my raw data in the form of a bar graph. And I've also added an excerpt of my genotyping data. Let's say I would like to notify members of a particular team. So as a review from last time, we showed how we're able to @mention individual users on Benchling, and they will receive an email notification as well as a link to that notebook entry in which they've been mentioned. But I'd also like to call out that you're able to @mention multiple users at once via @mentioning a team. So to @mention, we'll simply type in the @ symbol. And then rather than searching for an individual user, this time I'm going to search for a team, which in this case is the ELISA team. So I'll click on this option. And what this will do is this will @mention the admins of a particular team. So if in your organization, you frequently need to notify multiple admins that you have, for example, completed a certain step within your notebook entry, you're able to easily do this via an @mention to the team. Wonderful. So now that we've gone over, again, some of the features of the desktop sync application, as well as how to @mention a team to notify admins of a team that you've completed a certain part of your notebook entry, we'll now move forward with the witness and review program within the Benchling Notebook. So again, now that we've finished all the activities associated with this notebook entry, it's time to finalize this notebook entry so that it's all signed off and done. So in order to access the witness and review program, we can come to this review subtab within the notebook entry. And here we can see the status is in progress. This simply means that the notebook entry is being edited and that it's not sent out for review, nor has it been accepted. So this is the default setting for notebook entries, or excuse me, status for notebook entries when they are created. But now that I've, again, completed all of the activities that I need to do, I've checked off the protocol steps, and I've also added my external data. I'd like to send my notebook entry for review. Before I get started with that, I do want to jump back a little bit to settings and show how it's important to configure your project before you start using witness and review. So I'll again come to the projects application shown by the briefcase icon. And I'll want to look into the project settings by clicking on this gear icon next to the project, and then project settings. So we covered last time how from the project settings, we're able to see the different permissions of the different people associated with this project. And in particular, want to call out this auditor column. We can see for this particular project, we have set Benchling support as well as my colleague Stefan as auditors for this project. So importantly, only folks with the auditor box checked will be able to audit notebook entries or essentially retract or accept reviews for particular notebook entries. So I do want to give everyone a preview as to how this is configured so that you can configure your own projects for witness and review. So again, now that I've completed all the activities associated with this entry, I will now send the entry for review, which I'll do by clicking on this Send for Review button here in the upper right of the Review subtab. And we can see I've given the options to search for an auditor. In this case, it's already populated with my colleague Stefan. I do want to call out that if I come into this search bar and I search in Benchling support, I'm not able to see Benchling support. This is because by default on Benchling, we don't allow users to audit their own notebook entries. So if a user is set as an auditor for a given project, they won't be able to review their own notebook entries, again, in keeping with compliance rules. So again, I've set Stefan as the auditor for this project. I can leave a comment, which I'll do by simply typing into this leave a comment box. Wonderful. And now that I've left my comments, I can hit create review. So now that I've created review, we can see within the review subtab that I can see that I'm listed as the author. Stefan was selected as the auditor for this particular notebook entry. And we can see a log of the different witness and review changes that have been made. In particular, we can see that I made edits, and then I sent this entry for auditor review. We can also see a log of the comment that I left when I sent this out for review. I'd also like to call out that if I navigate back to the Note subtab, which holds the body of my notebook entry, we can see that I have this note that says editing is disabled because this entry is currently under review. This is because as part of the witness and review program, when you send an entry out from review, the entry is locked. We want to make sure that the auditor is confident that they are reviewing the finalized version. And I can indeed confirm that if I attempt to delete any contents or change any contents, I will be locked out from doing so. So while we are waiting on a few different steps, I do want to call out that the witness and review program does allow our users to maintain 21 CFR Part 11 compliance. So if this is relevant to your organization, please do look into the witness and review function. So I'm now going to perform a hard reset just to continue with our exercises as Stefan is deciding on whether he's going to accept my review. So I'll navigate to this review subtab. And I can see here that Stefan has retracted this entry. So essentially he has not accepted the review that I sent out, and he's added a comment to please add slide 2 of the PowerPoint. So I do want to call out that when you send an entry out for review, there is the option to either accept or reject the review. And now that the review has not been accepted, the review is again available for editing. So I'm able to make any of the changes that have been requested, and then I can again send it for review. So I was, in this particular case, asked to add an additional excerpt. So again, to review, to add an excerpt, I'll go to the relevant file attachment and click on the leaf icon. And from here, I was asked to add slide 2. So this case, rather than just clicking, I typed in the slide that I'd like to add. And you can also, of course, add multiple in this case, but I just want to add slide 2. So I'll click on Insert. And as you can see, we now have 2 different excerpts associated with this attached PowerPoint in my notebook entry. So I've now made the edits that were requested of me. I can come back to this Review subtab. And I can, again, confirm based on Stefan's comment that I have satisfied the asks for this particular entry. I'm also able to add an additional comment if, for example, I didn't understand the edits that were being requested. But in this case, they were pretty straightforward. So I'll hit resend for review. And I can add a comment as well. I'll hit submit. Again, the changes that have been made, as well as the comments associated, will show up in the log here under the review subtab. And if I come back to the notes subtab, again, because the entry is under review by the auditor, editing is disabled. I do want to also call out that the witness and review program is another instance when users will receive email notifications on Benchling. So if I come back to the review, both the author on this entry, which is in this case is Benchling support, as well as the auditor, all of these parties will receive email notifications whenever anything has changed with regards to the witness and review. So for example, sending an entry out for review generates an email, and then accepting or retracting a review also generates an email. So I'm going to give a hard refresh to see if my entry has been reviewed or not. And we can see here that the editing is disabled now because the entry has been accepted. So thankfully, I've made the correct edits and my entry has been accepted by the admins. And I can just confirm this by clicking on the review subtab. And we can see indeed that Stefan has approved my entry and believes we have some Nobel Prize winning research here as well. So again, to summarize, with the witness and review program, we sent out an entry for review, which locked the entry from being edited. And then auditors were given the choice to either accept the entry or reject the review. And then again, as part of the witness and review program, do want to call out that this will sync up with the home application. So I know we did go over this in the previous session, but the home application or the home icon is represented by the JeffyP logo here at the top of the left side navigation bar. And you'll be able to see a list or a preview of the different entries that are, for example, in progress or have been accepted. So I can, for example, click on where it says to accepted, and I can see a list of all the notebook entries that have been accepted. So I do want to wrap that feature up as well. Wonderful. So now that we've covered the witness and review program, which again is an important part of users on Benchling maintaining 21 CFR Part 11 compliance, we'll now walk through notebook templates. So if you'll remember from our notebook basics session, I previewed how when walking through the exercises of this particular notebook entry, how this entire entry was pre-populated as a template. So I'll now show how you're able to manage permissions and organize your templates and also to create new ones. So again, we're going to want to access the template settings on Benchling. And as you'll remember from both of today's sessions, the place to access settings is with the personal avatar in the lower left. So we'll again come down to our personal avatar located at the bottom of the left side navigation bar. And from here, in order to access the template settings, I'll come to Feature Settings. And from the resulting dropdown, I'll now click on Template Collections. So from here, I'm able to see any of the collections that I have for notebook templates. So in many ways, notebook templates, their structures and permissions mirrors what happens for notebook entries in projects. So rather than having projects, notebook templates have collections, and collections are a way for you to organize your templates and also set permissions for these templates. So in particular, we have this anthrax therapeutic studies collection already built, and I can also create a new collection by clicking where it says new collection. And now let's click into this anthrax therapeutic studies template collection. And we can see that we have a couple different notebook templates as part of this collection already. Importantly, we can access the settings for this template collection by clicking on this gear icon here in the upper right. And this, you'll notice, is very familiar to what you're used to seeing from the project settings. Again, we have the name as well as the description of the notebook template collection. We're also able to designate permissions for members of a team or for individuals on Benchling. So important, or one thing to call out is that permissions do work slightly differently for notebook template collections. You'll see here that we have, again, those same different options. And again, the none permissions is similar to what you remember from projects. So users with none permissions are not able to view these template collections, nor are they able to utilize these templates to create entries. One important difference that I'd like to call out is the read permissions. So users with the read permissions are able to view the notebook templates within a collection, and they're also able to create entries from any of these templates. But importantly, they're not able to edit any of the templates. So the read permissions in many cases is the most relevant for end users of your organization. If we have end users that don't need to be editing the templates, but will be utilizing them, we would recommend the read permissions in this case. We also have the write permissions. Users with write permissions for a collection are able to edit existing templates as well as create their own. And then users with the admin permissions are have all of the privileges of the previous levels of permissions, but they're also able to designate they're able to designate permissions from these template collection settings. So the reason I'm able to edit this is because I'm logged in as Benchling support and I'm the admin for this template collection. So I'll now dive into one of these templates. As you can see here, for example, we have the ELISA for bacterial toxin detection template that we used for our exercises in these sessions. So I'll click into here and you can see that essentially editing and creating these templates is exactly the same as working with notebook entries themselves. So essentially, if you know how to edit and create a notebook entry, you absolutely know how to create a template. Nothing new here. We have the same text editing and the same toolbars as well. Let's say, for example, I'd like to create a new template. I can do so by clicking where it says new template. I can have a name for my template. And then I'll hit create. And I've now created a new notebook entry template within this template collection. So again, I'm able to enter in any text. Some features that I'd like to call out for notebook templates. There's a few different ways that we can use templates. For example, we can put in different steps of a protocol. We can also add in tables. So again, as a review of last time, to insert a table, I'll click insert and then table. And I can set the dimensions of this table. So if you remember from last time, we're able to use these unstructured tables within a notebook entry in order to perform calculations. So from last time, we performed calculations on making a master mix. So here you're able to add a table and pre-populate it with the formulas you need to perform your calculations. So in this way, you'll be able to have this calculation table within all of the entries that you use from this template. Additionally, you're also able to add attachments directly to a notebook template. And this is done in exactly the same way as you would with a notebook entry, again, by clicking on insert and then attachment. One thing to call out here is that when you have a file attachment attached to a template, whenever you create an entry from that template, you will gain a unique iteration of that file attachment. So if, for example, you have some complex calculations that you need to be performing within an Excel spreadsheet, you can attach that Excel spreadsheet directly to your notebook template. And that way, whenever you create an entry from that template, you'll have a new unique version of that attachment that you'll be able to edit and perform your calculations within Desktop Sync, and then that will be synced up with that notebook entry. So again, highly recommend using unstructured tables as well as file attachments to really leverage the power of entry templates. And that brings us to our second poll question. So for our second poll, we're wondering, how could your teams utilize entry templates? So please do take some time to fill this out. Awesome, so it looks like everyone is filling out this Zoom poll. So I'll go ahead and end the poll. And I'll share the results. So as you're able to see, many of you said that you're going to utilize entry templates for data collection standardization, and that's certainly a great way to leverage this feature. You're able to put in areas where users can input exactly the data that they're collecting in a structured manner, such that it's standardized across your team. 29% of users or folks on this webinar also said they're able to use this for SOP creation. So again, I think this really touches on what we discussed in our last session, that we're able to utilize notebook entries as both observational records as well as for experimental protocols. So you can certainly do that with templates as well. And then 20% of folks also said that they will utilize these for team entry template sharing. So again, as we mentioned with the permissions that we're able to set for template collections, you can share your templates with any of the relevant members of your team. Awesome. So now that we've gone over some of the features regarding notebook templates, how we're able to specify permissions at the template collections level, and some of the main features that you may want to leverage when creating your notebook entry templates, let's dive into how you're able to find all these entries, whether you're creating them from templates or creating them from blank. So I'll now navigate to back to that main user interface by clicking on the JFEP icon. Because I do want to call out that the template collections, as well as some of the other settings on Benchling, do live in a slightly different user interface. So now let's say I'd like to search for some of my notebook entries. I can do so by clicking on this global search icon, which is represented as the magnifying glass here in that left side navigation bar. And as we can see here, I already have a search populated. Let's say this is not the search I'd like to go through. I can clear it by clicking on this gray clear icon. I now have a blank search. So global search is a powerful tool that allows you to have a high level of precision when searching for data on Benchling. And do want to call out that the global search will comb through any of the data that you have access to on Benchling. So importantly, it combs through data that lives both in projects, as well as if your organization uses the registry, it will also search through data within the registry. So we have a familiar keyword search where I'm able to search for a particular keyword. And then let's say in particular, I'd like to narrow down my search just to notebook entries. I can do so by clicking on the type dropdown, and then I can come down to entry. So I've now narrowed down my search to only notebook entries that contain the word Eliza. From here, I can also click on where it says folder in order to specify a particular project or folder location. Additionally, I can also add a filter by clicking on this filters button. And from here, I'll then click on this blue plus icon that says add filter. And I have a few different options for adding filters. For this example, I'll click on where it says name. And from the resulting dropdown, again, I have a few different options. I'll select this created filter. This allows me to specify a date of creation for a notebook entry. So let's say, for example, I'd like to look for notebook entries that have been created in the last week or so. So when I click into this from window, I get this calendar view where I'm able to select the initial date that I'm searching for. So let's say I'm looking at, I'll pick my start date as October 19th. And I'll then select today's date as the end date. I'll then click apply. So what this search has done is it's further narrowed down my search, which again is focused on this ELIZA keyword and then data of the type notebook entry. And then it's furthermore filtered my results to only show data that has been created within this time window, which in this case is from October 19th to October 27th. So again, you are able to add all of these different features iteratively. So I can add a keyword search and type, as well as multiple filters on top of each other. So again, we're giving you a high level of precision when it comes to narrowing down your search results. Additionally, an additional feature that I'd like to showcase on the global search relates to how the keyword search works. So I'll just clear this example search that I'm working with. And I'll now search this word synergy HT. And we can see here that multiple notebook entries have been are showing up here, including the one that I was just working on. So I'll click into it. As we can see here, it's already opened. I can click into this other one as well. Importantly, if I search for this word, just using a Control F search, you can see here that this word synergy HT is actually not located in the body of this particular notebook entry. But the reason that the Benchling global search has identified this notebook entry is because this string synergy HT is actually located in one of the attachments of this entry. So if I click into this indirect ELIZA raw data file attachment and look at the preview, we can see here that synergy HT was the reader type. So this was one of those metadata fields that was captured within this raw data file. So therefore, what we're showcasing here is that the keyword search that you can perform within global search searches not only the title as well as the body of a notebook entry, it will also search the text of different file attachments that you have for this entry. So this would be helpful if, for example, I had multiple different plate readers that I was working with within the lab, and I knew that, for example, the raw data that came off of these instruments always captured the particular instrument that had been used. I'm then able to perform a search via a keyword search to identify any of the notebook entries that are associated with data that came off of the Synergy HT plate reader. So again, I was able to comb through the file attachments in addition to the notes themselves. So this brings us back to our third poll question, our third and final question. And I'll launch this poll. So for our last question, we're wondering, without proper notebook searching, has finding specific notes been difficult for you in the past? So please do rate whether or not this has been a pain point for you. Wonderful. So thanks everyone for filling out this poll. So I will share the results. And as you can see here, we do have a spread of different responses to this poll. We can see 52% of folks felt like this was sort of difficult. So essentially searching for specific notes was sort of difficult. And there was certainly a range. Some folks have found this very difficult as well. So again, want to call out that utilizing global search, you're able to search for not only notebook entries, but again, any of the data that you have on Benchling, whether that is DNA sequences, or if your team utilizes the registry, you're also able to search for registry entities as well. So again, highly recommend utilizing global search, again, in lieu of utilizing a complex folder or project structure. Wonderful. So that brings us to the end of our exercises within the Benchling platform. Just to review what we covered within our exercises today, we covered how we're able to utilize the desktop sync app to maintain our file attachments as a central source of truth for our data within our notebook entries. We showed how we're able to make edits to our file attachments and have those sync up with the attachment that lives on Benchling. We also showed how we're able to add excerpts to our particular entries. We then dove into notebook template collections, where we're able to share templates with different team members, set permissions for those templates, and also create templates for standardized data capture, as many of you called out in the poll. And then to finish us off, we showed how we're able to leverage the global search to have a high level of precision when searching for notebook entries, again, as well as any of the other data on Benchling, with a particular highlight on using a keyword search to comb through text that's within not only the notes of our notebook entry, but also within the file attachments as well. So I do want to take some time to let everyone know about some recent product updates. Again, the Benchling platform is constantly evolving, and we are always looking to make new applications, or excuse me, to make new improvements. So I do want to call out that we do have these monthly product release announcements, which I know I previewed in the last session, but again, we are release these on a monthly cadence. And these will be emailed to admins of your Benchling organization. And you can also find these at help.benchling.com. And for example, we have the new release notes for October. Wonderful. So that brings us to the end of our exercises as well as our main content for today. So we would love everyone here to take a survey. So we are going to be sending out this survey within the chat. And so as, as this is our first Benchling Bootcamp series focused on the notebook, we do really appreciate any of your feedback. We're constantly looking to improve our training strategies. So please do take some time. This should only take a minute to complete. Wonderful. Well, thanks everyone for taking the time to give us feedback. Again, we really appreciate this, and we're looking forward to hosting more Benchling Bootcamp series in the future and constantly improving these series as well. So that brings us to the end of today's session. Do want to call out that we have some resources to share with you. Additionally, as part of our follow-up, for any users that were diligent enough to attend both the Notebook Basics session as well as the Notebook Advanced session, you will be receiving a certificate of training from our wonderful logo, Jeff EP. So do look out for that in your email. In terms of resources moving forward, if you ever need help while utilizing Benchling, please do leverage these Benchling help articles. These again are located at help.benchling.com. And this is an organized catalog of different help articles with screenshots and videos that help you to perform any functions that you need to perform within Benchling. If you need further support, we do have a couple of different ways that you can get in contact with us. You can utilize the in-app chat, which is accessed via clicking on this question mark icon, again, in the lower left of the Benchling screen. So do click there, and one of us from customer experience will get in touch with you shortly. And if you have more involved support needs, feel free to reach out to us at support@benchling.com. Additionally, this Benchling Bootcamp series on the notebook was an example of some of the trainings that we give on the notebook application, but do want to call out that we offer trainings on all of the applications on Benchling, and we can also offer custom trainings as well. So if your team is interested in further training, please do contact us at training@benchling.com. Again, all these emails and links will be sent out in a follow-up email. But do contact us there, as we can certainly connect you with members of the team who can give you additional training as needed. So again, to summarize, please leverage the help articles at help.benchling.com. If you need support, reach out to us via the in-app chat or email support@benchling.com. And if your team is interested in additional training or custom trainings on other applications, do reach out to us at training@benchling.com. Wonderful. So that brings us to the end of our session. Awesome. Was just making sure there. That brings us to the end of today's session. Thanks so much, everyone, for staying on this webinar and learning more about the notebook application in Benchling. So congratulations on finishing the inaugural very first Benchling Bootcamp session. We really appreciate everyone attending, and we look forward to your continued use of Benchling, and we look forward to getting in contact with you in the future. Thanks, everyone.